Hello students and welcome to another science online class. This is video number 10 for the topic biochemistry and biomolecules. Now we will continue our discussion about protein denaturation. So when we say denaturation, this is the loss of a protein's native structure. So what are the agents that causes denaturation? So it can be alteration in pH, salt concentration, temperature, or other environmental factors that can cause a protein to unravel. So a denatured protein is biologically inactive. So example is cooking our food. And so this is the normal protein after denaturation, which is the loss of biological activity, nagiging ganito, denatured protein. Pero pag nagkaroon ng um, regaining of activity, okay, ang tawag naman ay renaturation para bumalik sa normal protein. Yan. So, this is the native state, which is the egg. So, because of temperature, naging hard-boiled egg din. Ganun din ang pressure. Pero naman kapag nag-change in terms of pH, ayan. Example ito yung century egg. Denaturants, yung para mga substance or chemical substances. If you add to fresh egg, maging ganito. Interfaces or you use a whisk no sa kitchen. Ito kitchen equipment to. Yan, naging ganito ang itsura. Foamy, naging foamy. Now let's discuss nucleic acids that carries the genetic information. So there are two types of nucleic acids, the deoxyribonucleic acid and the ribonucleic acid, DNA and RNA. So nucleic acids are composed of monomers called nucleotides. Nucleoside is nitrogenous base plus pentose sugar, while nucleotide is composed of nitrogenous base, pentose sugar, and phosphate group. Pag kumpleto, nucleotide yung isang unit na yan. So the linkage that is present in nucleic acids are the phosphodiester bonds and glycosidic bonds. So this is the difference between RNA and DNA. So, di ba? Single-stranded ang RNA while DNA is double-stranded. So, they have differences in terms of nitrogenous bases. Okay, so RNA has cytosine, guanine, adenine, and uracil, while DNA ay may cytosine, guanine, adenine, at nagkaiba sa thymine. And remember, they are different in terms of sugar. Yung carbohydrates nila, RNA is ribose, ito naman, deoxyribose. So this is the sticky sugar plus phosphate and nitrogenous base. Drop the base. Diba? Ang cute. Ulit. Sticky sugar, a phosphate group, and base. Drop the base. Lahat sila. So this is the DNA, no? So DNA structure that compose the chromosome itself. Yeah. So this is the nucleotide structure, phosphate group, the sugar, pentose sugar, and the base. Ayan. So again, adenine and guanine are the purine bases, while cytosine and thymine are pyrimidine bases. This is in DNA, while in RNA we have ribose, yung sugar, and one of the nitrogenous base na present sa RNA ay uracil. So DNA, yan, thymine. Sa RNA, uracil, ang nitrogenous base. DNA and RNA, present ang adenine, guanine, and cytosine. DNA and deoxyribose, while RNA is ribose. Present for both DNA and RNA is phosphate. So this is the difference between RNA and DNA.